Hey, science research. Welcome to another week of online learning. I hope you guys are all doing well and your families are safe and healthy. Um, if at any time you have a concern, whether it's a health concern with your family or there's something going on that you're not sure about getting the work done, just reach out to me. You know I'm flexible. This is a really crazy, critical time in our lives right now. Um, your life, your family, the needs of your families are priority one. Okay, school comes second. We can always figure something out. So always remember to reach out to me. I miss you guys. I haven't seen you guys in a long time, and I had so many really cool things I wanted to do with you guys, and well, here we are. So this week we're getting into some of my favorite stuff. This is JMol. Okay, so some of the stuff that we've talked about in the past with proteins, understanding protein structure, understanding alpha helix and beta sheet and all that was just to kind of get you guys warmed up to what you're going to do with this. This is the exciting stuff for me. So the first thing I'm going to ask you to do is to open up this JMol tutorial and download it onto your computer. Okay. The links don't work in Schoology, so you have to download it. So download it as a PDB, as a PDF file. Okay, you can put that on your computer. What I would do is put this on your desktop. So find a nice clean spot on your desktop. This way you can put this on there. Okay. What I want to do is just spend a few minutes just showing you some of the functionality of this program and how to get started with the tutorial. You are free to move through everything on this page at your own pace. Um, what I'm going to ask you to do is to go through the tutorial first, and then the rest of what we have in here, we're probably going to be doing this next week and even the week after. Some of you are going to move through this a little bit faster than others. That's cool. Go at your own pace, okay? This is an elective class. I know you guys have other classes and things that you're trying to prep for. So I want you to use this space as a place to just kind of play around and try things out. Of course, I do want you to get these done, and we'll get into that in detail once we kind of get moving with this. Okay. So in the meantime, hopefully you have the tutorial downloaded, and when you open it up on your um, desktop, this is what it should look like. And the first page is how to download JMol. Okay, JMol. What JMol actually is well, it says it right here. It's an open source Java viewer for chemical structures in 3D. It's a freeware that allows scientists all over the world to visualize and view different types of structures. We use it to look at proteins, but you can use it to look at all kinds of things. You can look at DNA, you can look at RNA, you can look at organic compounds, inorganic compounds. And the easiest way to get this is to download it. And so you go to this link here, sourceforge.net. You click on that and you can download it. It's a safe program. It's a JavaScript program. It's constantly being updated. And so if you scroll down on this page, you'll see where the download is, okay? Or you can just click the download button, okay? And this gives you the information on how to download JMol. So if you click up here on download, it takes you right to the download page. Click on this link. That is the direct link to download it. It comes in as a zip file. You're going to need to unzip it. And then once you unzip it, you're good to go. So if you read these instructions here, it says installing and running JMol. You only have to extract. This is important. Once you unzip it, you only extract this jar file from the download page. Put it on your desktop. Okay, that's the easiest place to put it. When you download, I'm sorry, when you double click on that icon, it'll run. Okay, so once you've downloaded it, just take that jar file and put it somewhere. I put it on my desktop. Okay, and the instructions on what to do are right here. Okay, so locate the download file in your downloads folder or wherever download files are found. It's a zip, and then what you want to do is put it on your desktop. So you'll get a file that looks like this. All right, jmol.jar. I'll move this so you can see it. It's kind of light up there. I'll put it in the water. That's what you're going to use to open up JMol. Okay. Now, what does JMol do? JMol reads what are called PDB files. Okay. A PDB file is a protein data bank file. That is a file that has the structure of a protein taken from X-ray crystallography. Okay. So that file is uploaded by scientists or ESM students who have identified and completely refined the structure of a protein and now want to share it with the scientific community. So the protein data bank was originally run out of Brookhaven National Lab, and now it's run somewhere else. But it has over 160,000 biological macromolecule structures. So structures of DNA, RNA, proteins. Mostly proteins are on here. That's why it's called the Protein Data Bank. And what people do is they download and deposit, or upload and deposit, the protein file so that other people can use this to advance science. It can be used to develop medicines. It can be used to develop drugs that treat certain diseases. Okay, and so when you go to, if you click on this link, rcsb.org, it takes you to the Protein Data Bank, and here we are. Okay, Protein Data Bank has lots of things on it. It has molecules of the month. Yes, they talk about different molecules each month. But basically, it's a free repository of protein structures. So for example, ESM students Chris Janata and Damien Adele deposited a structure. That structure was designated with this ID number, 6UEV. 6UEV was a number given to the protein structure that these guys worked on. So if you type that in and hit enter, it pops up, and here it is. Okay, you get a screen that gives you a picture of what the protein looks like and some information. The title of the paper that's being written, who were the people that worked on it. Okay, here I am. When this was released, what organism it came from, what is the molecule called, and what is the unique ligand. GOL stands for glycerol. Now, if you want to get more information on the protein, what you do is you click on the title, and now this opens it up to our specific page. Okay, and this gives you all the information about it. Look, it shows us the experimental data snapshot. It shows us what our resolution was. How good did our protein come out to be? The paper, which is yet to be published, but will be published. If you go up on the top here, this gives us information about the experiment. Okay, and if you click on experiment, it tells you what they did. Okay, and what the details were. It tells you about the crystal, what the crystal looked like. It gives us information about the diffraction data. What was the radiation source? Oh, this was from the NSLS2 beamline. It was a synchrotron. Okay, it talks about our data collection, refinement. So there's a lot of information in here that tells you what we did on our protein. This shows the software that we used, MolRep. Anybody that was, and RefMax. So if you were in that data analysis workshop we did, this is what we use that for. We use it to refine the structure. Okay, we go back to structure summary. Here's our protein again. Okay, here's the assembly of what it looks like. So we can click on this and get a 3D view of it online. Okay, it's loading up. Ooh, I clicked too many times. There we go. And so we can kind of see, here's our sequence. This is what our protein actually looked like. Let me bring this down a bit. So you can see we can rotate this around. Notice the alpha helices, right? We have tons of alpha helices here, okay? We have some beta sheets. See the beta sheets, these flat sheets, okay? This is, if you notice that these kind of look like mirror images of each other, that's correct. This happens to be a dimer. A dimer is a molecule made up of two similar molecules with the same exact molecules, okay? So we have one dimer here, and here's another one, okay? And this protein operates as a dimer, and we can see right here, you see where those glycerols are? I'll zoom in. That's the active site, okay? So you can see where this protein has an interface where the two dimers, the two halves come together, okay? Ooh, look right here, you can see this. Look at all these beta sheets. See how they crisscross each other? Okay, it's pretty cool looking, okay? So anyway, so that's that protein, but I want to get back to the tutorial so that we can go through this a little bit. So what you will do is in the search box, you're going to type in 1AL1, and that will allow you to see that particular protein, okay? This is the structure summary that you should get, okay, once you click on the actual protein itself. And you can see the X-ray diffraction data of this. This is called the crystal structure of alpha 1. This is an alpha helical molecule. Okay, this has information about who deposited the data and the date the data was deposited. It gives you some information about the resolution. The PDB validation is really important for crystallographers who are interested in the quality of the structure. How well does it fit the data? How are the bond lengths and angles? Do they conform to standard values? Is there clashing? Then there's a citation and an abstract, which tells you the journal that it was published in, right? And that's the literature. Okay. Now, what we're interested in for proteins is also looking at the sequence. Okay. Um, so in macromolecules, this gives us the amino acid residue sequence, right? So this is made up of 12 amino acids. This is real tiny. Okay. ELL, KKLL, EEL, KG. You notice the E's, the L's, the K's, L's, E's, L's, K, and then a G. Everything is repetitive in there a little bit, okay? And also it's using the same amino acids, right? There's really what, 1, 2, 3, 4 different amino acids in there. That's it, okay? There's a small molecules box uh, which tells you about anything that's non-amino acid, right? There's a sulfate ion in there, okay? Tells you what else we have, some kind of non-polymer molecule, okay? So the ligands just tell us what's in there. This is an acetyl group, okay? So the ligands tells us that a sulfate ion is in there. And if you look at the structure, all right, there's something right here, okay? So that tells you what that is, okay? So if you want to study this, what you do is you download the files, okay? And you click on download files, you're gonna get the PDB format file. Okay, that's what you want to download. So let me go back to that. L1. Ugh, allergies are kicking in. Here's one L1. Download the file. So I'm going to download this file, PDB format. Now, really tricky. When you download this file, let's do this again. It's going to come down as, I downloaded 2 of them, as a PDB file. Now, you can't open this up. I'm going to show you. If you try to open this, this is what's going to happen. Nothing. Okay. Computers can't read this file. You have to use JMol to read this file. So here's what you're going to do. You're going to take this file and you're going to just sneak it right over here onto your desktop. Okay. And you notice how it's got this blank look on it, right? Blank file. Okay. You have to open this file using JMol. Here's what I recommend you do. You create a new folder on your desktop called JMol. Okay. And all of these files that you download from the PDB go into JMol. Okay. Now my computer's running a little slow because I'm running the screencast on it. So it's kind of slowing me down a bit. See, I need an app to open this up. So I'm not going to use the app from the App Store because we already have one. It's called JMol. So I just want to let this go away. And when that goes away, here it is. Okay, so that's my JMol folder. And here's my one AL1 PDB. Okay, so I know what's in there. Okay, so now we want to open up JMol. Okay, you open up JMol by double clicking that jar file. Right, so there's my jar file. Okay, and then in the file menu, you're going to click on open. Okay, so I'm going to show you guys how to do this. So here's my JMol opening up. Okay, I'm going to move my JMol over here just so that I can keep the instructions open with you guys at the same time. All right, so now you're going to go to File, okay? Moving slow here. Okay. Go to File, Open, okay? And it's very slow. I'm going to look on my desktop. So you can click on that little window up here and you can find your desktop. So I'm going to my desktop and I'm going to go to JMol. And here's my PDB file. Now, once I click on that PDB file and hit open, boom, looks just like what's in the tutorial. Now, in order to work with this, first you can put your mouse over this. You can rotate this around. You can see that there are all kinds of molecules in here, okay? And this thing looks kind of like a ribbon, right? They call this a ribbon or a cartoon. So what we want to do now is open up our console. So to do that, you're going to click on File, Console, okay? When this opens up, you get this window with some information about it. But what's important is right down here, the red $sign. That's your command prompt. That's where you're going to enter commands. Now, there are a whole bunch of commands for JMol. There's a JMol command sheet. Let me just go back to this for a minute. Here's a JMol command sheet. You could open this up, download it. You can play around with this and just type in the commands and see what happens with them. It tells you exactly what to do and how to use them. It's a really nice, colorful command sheet. And eventually, you'll just become very comfortable with those commands and know exactly what to do with it. But for this, what you want to do is take your console and set it up so that these are side by side next to each other, okay? So that you can see what you're doing in the console while looking at your protein, okay? What I'm going to do, though, is just put my console here so I can follow along with you guys. And I'm going to just take you through these first few steps, okay? So there are a couple of things that you can do. You can manipulate the molecule with your mouse, okay? So again, you can rotate it, you can move it about, okay? You can do different things. If you hold the shift key down, so I'll hit shift, right? I can now left click, I can zoom in and out, okay? We can rotate, we can use our roller in the mouse. So if you have a roller on your mouse, you can roll to zoom in and out. You can hold the mouse down and zoom in and out by pushing down on the roller. Okay, so there's a lot you can do with this as far as just like manipulating it. And again, play around with that, try it for a little bit. So once you have this all set, what we want you to do is go through this. This is a step-by-step process for teaching you how to go through this. So take your time with this. Now, I just want to show you, let's say you do a few things. I'm going to just type in a couple of quick things. So hang on a second. Let's see. Here we go. I just did some playing around with the protein, right? So now I have all these weird things on it. Don't panic. You're going to learn what all those are. Let's say I've done this for a little while. I'm getting tired of it. I want to save it. Okay. To save your protein structure the way it is, you're going to click on this little camera icon. Okay. It says save current view as an image. When you do that, it's going to ask you where to save it. Make sure you save this in your JMol folder where that PDB file was that you downloaded from the protein data bank. All you need to do is click save. Okay. Now what does that do? Okay, let's go to the, I'm going to close JMol. I'm done. I saved. Okay. Now I want to go back into that. I just want to show you something. You save that as an image. So right here, it saves it as a JPEG. That is the still shot of that protein. And it saves it as an image, but it also saves all the parameters of what you put in. Okay. So yes, it's saved as an image, but it also contains all the background coding necessary for you to see that structure. So when this image comes up, it saves it as a picture, and it even puts a little JMol icon on the bottom. Okay, so that's the image. Now, if I wanted to, let's say that this is a protein I wanted to put on a display and do a presentation, I can take this and put that on a poster, and there's my picture, or put it into an article, and I've got my picture from JMol. Okay, but what's important about this is you want this to stay in the folder next to the PDB file. Okay, here's why. These talk to each other. Okay, so when you go to open this up, let's say it's 2 days from now, and I was like, oh, I want to go back and work on my protein. I will go to File, Open, or just go to my Open, right? I'm going to look for my desktop. I want to go to this desktop, JMol. Here we go. And I'm going to go to that JPEG file. Right, here's my JPEG. Watch what happens. When I open this up, poof, it's that image. Now, if I turn it this way and save it, it'll save it in that orientation. So what's kind of neat about this is if you're going to do a presentation and you want to save your protein and focus on something, like let's say I wanted to show, the loop here, okay, I could do it this way, save the image, save it as a JPEG. Do I want to overwrite it? Yeah, because I did something different, right? And now if I close this and go to look at that picture again, okay, hopefully this won't take so long. It'll be in the same orientation that I just put it in, see? So you can really play around with this a bit and manipulate it. But what's important is whatever changes you make sure it goes in the folder right next to that PDB file, okay? So that you can play around with it, all right? All right, so back to this. What you're going to do is go through the instructions for the script console. Take your time with this, okay? If you mess things up, don't worry. Just hit undo, okay? Um, what I'm going to do is just grade you on completion, okay? I want to just see what it looks like, so don't panic. Play around with this. This is something I want you to just explore and learn about. It's a really great program to use, okay? Now, the one question people have here, I even put the information in here that this is what it should look like as you're typing stuff in. So I even did this to show you some things, okay? Look how pretty that is. Wow, see? That's the same protein, right? Pretty cool. But here's what I want you to do. At the end of this, once you're done, what I want you to do is take a screenshot of it or just email me the image. I just want to see what it looks like, just so I can see how it works, okay? What we're going to do is build you through more complex structures as time goes on. And eventually what you'll wind up with is a structure that, is really complicated. The rest of these on here are little tutorials, right? So like zinc finger tutorial, if you click on this, it literally just gives you the commands. All I want, you don't even have to download this. You can actually go right off of this and then just open up JMol. Literally want you to just go through these commands and just see what they do. Okay, that's what this is about. This is about learning and seeing what these different commands do. Okay, there is a lot in here. Don't expect this to be done in a week. I want you to move at your own pace. If you get stuck, let me know. We have those little extra help sessions, those office hour sessions that have been set up. So I'll be around. So if you're stuck on things, we can screen share and look at the programs, and I can kind of tell you what's going on if you get stuck. Okay. But again, something to just play around with. nothing written, nothing that you have to submit other than I just want you to take a screenshot of what you have, okay? So again, go through this. This is a long tutorial, okay? It's going to take time to go through. Originally, this was stuff that you were going to do in class, okay? Um, but again, you're going to look at this on the proteins that you're working on, see? And you're going to play around. I made screenshots of this too, so you kind of get the sense of, oh, this is what things should look like as you're doing this, okay? So I went through this myself. I'm 95% sure I got all the little kinks out of it, so you can see how things work. And then we'll go through it, all right? But there are lots of exercises in here to go through. You can see this is long. Again, don't rush, don't panic. Um, really, I'm thinking this is probably 2 weeks worth of work. But hey, you know what? If you move through it quickly, more power to you. You move through it quickly, okay? You guys could work on that this week. Just spend 2 days on it. That's all. Spend about, you know, 45 minutes to an hour, twice this week. Do a little bit more next week. And then what I'll do is we'll check in. We'll make sure that everybody's doing okay. And, if you're done with it, move on. Start looking at those other tutorials. Okay. So that's it. I don't want to talk too much longer because this video is getting way too long and I don't want to bore anybody. But again, guys, I miss you. Stay well, stay healthy. And again, just shoot me an email or remind if you need anything. And I'll help you out as best I can. Okay. Have a great day. Be well. Bye, guys.